So good morning, everybody. I think it's time to start. Welcome to our faculty colloquium. Today we have a guest from the neighboring institution, which is separated from us with a solid fence, and it's a Nencki Institute. And Dr. Pawel Boguszewski is doing research, preclinical research, mostly on mice, but also on fish, I guess. But today he will speak about the consciousness as a natural phenomenon. Please, Pawel.
Good morning. Thank you for coming. It's Monday before the noon, so I really appreciate your presence here. Some people are nodding. Okay. So today I want to talk about the something which is not a subject of my direct research, but it's something that I think it's really worth to think about and consider it. So consciousness. So just imagine a situation that you have to go to some surgical operation, nothing serious, but you know you're going to be put in a full anesthesia. The doctor is using three types of drugs. The one is to put you into the sleep, so you don't have to remember all these, nasty sounds and, talks. Then it's something to make you paralyzed, that your muscles are not twitching. And the third one is painkiller, because even when you are sleeping, you can have some reflexes. And if you are not very lucky, and the chance is 1 in 20,000, you can wake up during this surgery. And after this talk, you will know that the anesthesiologist is not able to recognize that you are awake because you are paralyzed. So the consciousness is something which is really like a huge topic in philosophy, but also it got really interesting connotation to the everyday life. So this not accidental awareness during the surgery is something which really ties it to the everyday life. All right, but as I said, the consciousness is something which is the biggest question which is before the science, because this is something that connects our feeling of being self, our, let's say, experience of world with matter, with something that should be common for everyone, that we can investigate. And if you ask me what is the biggest question before the, let's say, humanity, that's the one. I think there is no bigger than, even the origin of universe is less interesting than the consciousness. All right, so I want to start with describing what I will talk about. So it's going to be like in a 7 short chapters with different length. So I would like to start with the brain. And why? Because, I'm a neurobiologist, so the brain is the most important thing in the universe. And as we know, this is the device that is producing the consciousness. If we take a look at the brain from the biological point of view, we know a lot, but still there is a lot of mystery and things that we don't understand. So, starting from the beginning, this is a product of natural evolution. Means on this planet we got something strange which is called life. The life produces very complex chemicals, and those chemicals turn out to be us. And each brain is shaped by two big forces in a biology. The first one is genetic influence. Means if you take a brain of the Homo sapiens, you know the parts, you know how it's built. And with our brain comes not only the build, the hardware, but also a lot of software. So in our genes we got encoded things like preparedness for language, face recognition, and also, for example, the proto-morality that serves us on the very beginning of life. So it's not something that it's given by the culture, it's innate. But on the other hand, each brain is different. Even if we take identical twins, the genes are 100% identical, their brains during the development will differ. And also through all of our life, we are combining the experience, the learning, which is a plasticity, with some mild damage, and all our life makes our brain different. So the problem is that the subject of this part of the talk is something which is really unique from human to human. When we take a look how it works, and we got a couple hundred years of trying to understand how the brain works, I think the most important part is the last 120 years. So now we know, first, it's very not energy efficient. It is comparing to other devices that we are producing, but it consumes up to 25% of energy that we are taking in our body, and it comprises just 2% of our body mass. So it's like a 10 times more energy consuming device than the other tissue. So probably it's useful, because in nature something which is not useful is just eliminated quite fast. It comprises of the cells as everything in the living world, but those cells are very specific. We got in our brain around 86 billions of them, so that's quite a lot. They tend to have a special shape, means the cell body, because you need all these, cell guts inside to just keep working. But what is characteristic, they have extended like the dendrites, which are for collecting the information, and axon that just sends the information to other cells. And they make connections, synaptic connections. There are around between a couple hundreds up to 10 of thousands connections between each cell. But what we know right now, that they are working with electrical impulses, means there is nothing special, like no specific phenomenon that let them work. It's just electrical impulses. And also when they talk to each other, they use the oldest way of communication in living systems. It's a chemical signal. So just being more specific about these electrical pulses, because that's a proper place to talk about the electricity. We can see even on a single cell that it's not like one impulse. There are many different impulses. For example, the cell, this is a pyramidal cell from the cortex. We can see the small protrudings over here. There are the synaptic spines. I will talk about them later. This is, let's say, input. So the cell is just producing a special place, saying, okay, I need a connection here. And signals that are obtaining through these spines are averaging over the dendrites and cell body. And when those signals cross the special threshold, the threshold is changing in time, and it can vary between the cells. The cell is producing a single output, which are the spikes, like a firing. So what we can see, the single neuron, and as I said, we got 86 billions of them, is a quite complicated signal processing unit. And then when cells talk to each other, they use special chemical impulses. Okay, so that's them. So that's why we need a new type of, let's say, science to research this. So that's why right now we talk about we don't talk about the biology of the nervous system. We rather talk about the people are, let's say, gathering under the umbrella of neuroscience. Because this is the approach that we adopt to try to understand the brain. I mean, like, let's ask any type of, let's say, already established scientific branches what you can say us about the brain. So right now, when we just take a look at the neuroscientific community, you can see biologists, of course, because that's a biology rooted problem, but also we got like philosophers, psychology, computer science, especially computer science, psychologists, chemistry, and so on. And this interdisciplinary approach is really seen when we just take a look, for example, at the last year Nobel Prize. So we have for Hopfield and Hinton the first award. It was from for physics achievements, and this is about the neural networks. So something that was really roughly inspired by how the brain works. And what they try to do is they just model the connectiveness between the cells in a computer, and they just develop the science of neural networks, artificial neural networks. And their work is quite, let's say, old, because it's from, I think, 60s. And exactly, even those days we could read in a Młody Technik. Probably majority of you knows the feeling of buying a new edition of Młody Technik and just going through and, for example, reading the Professor Tadeuszewicz articles about the neural networks, and that was exactly something that inspired me to go to the biology and also keeping my eye on the computer science. All right, but of course, in biology, if something can be more complicated than it should be, it will be. So it's not only about the signals, about the electrical pulses, which are really, let's say, elegant. We can measure them, we can trace them. Of course, there are a lot of them, but still, we can manage them. Because in biology, we also got this layer of, let's say, chemical interactions. So when the cells are talking to each other and all, let's say, weight of connections are hidden in a synaptic connections, we have different type of information transmission. So this is the ending of the first neuron. That's the, let's say, ear of the second one. And here the electrical pulse is producing outflux of the chemicals, neurotransmitters. And the efficiency of transmission of the information depends not only about the electrical signal, but also about the amount of chemicals that are released, and especially about the proteins that are able to listen to this signal. So that adds another layer of complexity. And we see exactly that this is on the border of chemistry and the biology, because the proteins, we cannot say they are living. They are organic components, but yeah, it's just a name. So they are exactly, let's say, sitting between the life of chemistry and something we can see in a living organism. So we got these proteins, and in our brain, the most important receptors, the proteins that are receiving the chemicals and producing postsynaptic currents, are AMPA receptors and GABA A receptors. So right now we are listening, seeing, paying attention exactly using those two chemicals all the time. And the ions that are used to produce the currents, it's sodium and chloride, and this is just a salt kitchen. So right now we are processing the information using a salt kitchen. All right, and this is almost the lower level. And then this chemical world keep getting complicated, because we got these compounds, chemical compounds, that are using the proteins to produce the electrical impulses, but also we got the whole apparatus, like we need to produce those proteins, we need to produce those neurotransmitters, we have to transport them, we have to reuse them. Like, for example, in reuptake, which is the basic of many drugs. And then we have the whole brain, means those cells are in a particular places, they differ with their content of receptors. So the whole picture is much more complicated than just electrical signals in the brain. And even if you take a look at the circuit level, because the single cell is complicated, but the circuits are much more complicated. Right now we are thinking about bigger units in the brain, like cortical columns. And this is small ensemble, small, because there are like 150,000 cells, different cells, that are just forming a single place in the cortex. This column is the roughly size of the rice grain. And right now we are thinking that this is like a very basic computational unit, and our brain is smart because we were able to multiply these units in our cortex. And to make the things more complicated, we developed new techniques. For example, this is a very cool technique that you can take a look at the brain of the mice through the specially designed window, and you can use a microscope that will track the neurons over the time, for example, couple weeks, because you just scan the mice every couple days. And what researchers realized that this network is not a static one. We knew that we got always new connections because that's the basic of the memory. But what they were surprised that we have a very huge turnover. Like in mice, 40% of connections were destroyed and renewed within the 4 days. So it's not like a static device. It's changing over and over. And if we take a look at how it really looks like, because on this schema it's really looks like a very straight neuron connections, really beautiful. But the real picture is like this, and this is a very new picture from Nature article where researcher produced the first whole brain image with each cell. This comprises of 150,000 cells, and it's from the fruit fly. So that's let's say the biggest brain we scan completely from top to the bottom, and they calculate exactly 139,000 something cells with over the 50 million connections in such a small brain. They identify more than 8,000 types of neurons, and 4,000 of them were unknown before. They didn't got the idea that there exist. What was really interesting about this paper is that when you just imagine this fruit fly, the organism that is just hovering over the rotten apple at your home. Okay, so if you just take a look at this organism, it's really smart because it can navigate in 3D. It can memorize information. It learns. It reacts to your hand, waving around the apple. They also got social interactions because they have to mate, and they can also sing to females. So just 150,000 neurons and so complicated behavior in a brain which is very, 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 very small. What was interesting about this paper is that they use artificial intelligence to track all this, the mess, but they also build a game. So people were playing the game, just coloring online the neurons, correcting the algorithms. And the best player was from Poland, from Katowice, and they call it like a crowd science. So you have a lot of tasks, so if you just make a game, you will always find people who like to play games. So you can go to the iWire.org and you can just, that's my account. I just had to check how it works. All right, so this is, let's say, how we think the brain is working at the bottom, but on the top, we still have no idea how the whole brain is what is the role of the whole brain? For many years, we thought it's a special device which is just reactive. Means we present it with a stimuli, for example, strawberry, and the task is just press the button when you see the strawberry. Okay? So, because, as I said, there are electrical signals, if we just don't think about the chemical part, it's quite easy to measure. So we can just measure the transmission of the signal through the visual system, which is quite complicated, because our visual system comprises of many subsystems that are just building the image. So we end up that the reaction, the physical reaction, the movement, is around 180, 200 milliseconds. So that's a lot, 1/5 of the second. Playing tennis is not possible at all. You cannot play the tennis. Why? Because even if we think about the visual system, and the slowest part is retina, because you got chemicals that should be saturated with photons and then produce this electrical pulse, it takes at least 40 milliseconds. So, very basic math. If you just take an average speed of ball, usually you see the ball half meter after it is in reality. Okay? In case of very fast servings, like I think the record is 263 kilometers per hour, it's 3 meters. Okay? So you cannot see the ball. It's not possible. So right now, and of course, I show you just the one pathway, but in case of visual system, we got at least 2 or 3 systems, because you have units that are processing what you are seeing, and this is this part, and automatically on the, let's say, ventral side, and where, and if it moves, so that's a dorsal part. And then the brain has to just combine all these streams into the single picture, like a moving picture, and of course it's waiting for the slowest department, okay? So it takes a lot of time. So right now we know that it's rather prediction and correction of errors than just really reacting to what we are seeing. So we are seeing the ball in our head because we know where it should be. That's why the training of the tennis takes a lot of time, because it's not about the physical. It's about having the best prediction. Okay, and we can see that in visual illusions. Okay, so just take a look at this picture, and this part over here, if we just measure the intensity of light with some device, it's exactly the same like here. Okay? Yes or no? Maybe. Okay, good. Yeah. Physics department. All right, so I saw this picture 1,000 times before, okay? And my brain is still forcing me to believe that here is a dark gray and this is a white, which is not. Because if we just put something like bar over here, we can see it's exactly the same, okay? Oops, sorry. Oops, not that, okay. So why, when I'm just flipping it, something is right. So why it's so? Because our brain knows that it's so slow, so we have to predict how the world really works and then just add, let's say, error correction. So in this case, the brain is just building 3D picture because you know that retina is flat and the world is just, three-dimensional. So probably the light comes from the top, like lamps or the sun, and it's just forcing you, because something which is in the light should be darker if it sends exactly the same of our brain. And we got a lot of such a prediction. I already showed that picture because, device malfunction. So researchers were interesting how our brain is really relying on a prediction, or on, let's say, I will see what I want to see or what I have to see. And in this case, we have the morphed picture of the face, like 50% Clinton, 50% Bush. And if you present original picture of one of the presidents and then show this ambiguous picture, your brain will see the opposite signal, okay? So you have a situation when you just present exactly the same picture to two different groups of people and they will see a different person, okay? So this is not that they perceive the information from the picture, they also use the memory a short time, like just seeing the original picture for the 4 seconds, but also they can use like their attitude, okay? Who I want to see, okay? I'm a Democrats, I will see the Clinton. I'm Republican, I will see the Bush, okay? All right, so theories. So we as you can see, we got a lot of problems with the brain. We know a lot, but still a lot to go. All right, so the consciousness itself, I tried to find out the definition because I like definitions, and I was able to find hundreds or even thousands. And when we got many definitions of a phenomenon, probably we have no idea what the phenomenon is, and that's exactly the case. So, well, probably the problem is because the word consciousness came from the, let's say, pre-scientific era, so we got a feeling what is this, but then we can talk about some aspects. Probably the same is with energy in physics, okay? In case of my work, it's also emotions. Like in emotions, we put fear, which is a very, let's say, straightforward emotion, but also a catharsis or love, okay? That's exactly in the same bucket. So probably with consciousness, we got exactly the same case. But this is not only a problem with definition, but also it touches this mind-body problem. And the most prominent example is Descartes, who thought that, okay, we know that we are, and he got this idea that we got a continuity with animals. We are not a very special entity in the world, that we got humans, animals, plants, and the rest, but we are the part of the animal kingdom, but somehow we are special. And he coined the term cogito ergo sum, that what we can be really sure that is that we are thinking, okay? So for sure I am. I'm not sure about you. But also he introduces the dualism. And that's a problem because for up to date, we got people who are thinking that, for example, animals are just machines, proteins machines without no, let's say, conscious processing. And this idea of dualism was criticized very quickly because you can read the book, which is the correspondence between Princess Elizabeth of Bohemia, and she was a young lady. She was really criticizing his idea because he said, okay, we have this, let's say, part which is material, and we have this part which is purely spiritual, and they cannot interact, but somehow they interact. So, all right, so right now we are standing as a scientist on a view of monism. Physical monism means that we think that consciousness is a product of the brain, of the matter, until we will have another proof. But still, the debate is from views like, for example, Dennett, that consciousness is something which is a concept like that's cool, that's fancy, okay? So we just call some phenomenon which is virtual, and he names that consciousness is something like desktop in a computer. It's not existing at all. It has no substrate. It's just some kind of process that is helping us thinking. Or, for example, Sir Roger Penrose, Nobel Prize laureate, who is advocating that we cannot know what the consciousness is because it's a non-computational problem, and its solution lies in quantum mechanics. And as far as I know, no biologist is supporting such a state. For sure, we will have one problem, But I think we are still far away from reaching this threshold. David Chalmers coined the name of hard problem. That easy problem with a consciousness is how we process the information, okay? How we are conscious that we are seeing something or thinking something, and that's, he named it's easy, because we can just look for the correlates in the brain, and maybe we will find out how it works. But the really hard one will be, we will never feel how it is to be another person, whether another person got this, let's say, unique feeling of being self, okay? This is something that we can talk about, we can say someone about our experience, but the second person will never feel that, okay? So it's, this inner experience is something which is, which maybe will be this threshold that we cannot cross. But anyway, on the way, we can just try to figure out how it works. So in the 90s, Crick and Koch, Crick is the guy from DNA research, they thought, okay, so if we just take a look at the theories, thousands of them, okay, if we take a look at the different, approach, many, so let's start without the theoretical background. Let's just start tinkering, like looking for correlates in a brain. So what they were talking, it's not to design the theory, but just let's look for the neuronal correlates of consciousness. And the framework is like this. We can present with some stimuli, ask persons or force them to, for example, feel or think about something, and then let's try to measure the neuronal activity on a different level, electrical, chemicals, so on, and let's try to combine whether we can just find out something. So I really like this approach because it's like a more engineering approach than the philosopher's approach. But it's more true to the fact. But the problem is that we still end up with at least this new paper 2022 by SET. They just combine all the theories that emerge from such experiments of looking for correlates, and there are at least 22 of them. I will just show you 3, which is the first is the higher order theories. So they think that we should have in the brain a special place, a site of consciousness, which is separate from other parts, okay? So like supervisor, okay? And this supervisor, this homunculus, is sitting somewhere in the brain. We should be able to find this process, and is just taking a look at the, let's say, unconsciousness processing of information, for example, like a visual information. So I'm seeing something that produces some activity in the brain, and then we have a separate entity, and this entity is looking at this activity, okay? We got another set of theories, global workspace. It says there is no such a place, there is no a particular place in the brain that is looking at the other places. We have just like a global workspace, means those non-conscious places that are processing information, they are just combining together, and if you got a lot of them, then they somehow produce the consciousness. In this case, they claim that consciousness is just a numerical process. Means that maybe if we can copy such a stuff into the computer, then we will be able to produce a human-like consciousness in silico. Okay? In opposition, we got integrated information theory, and they claim that the consciousness is something really like a fabric of universe, that each entity got some small consciousness. If you have a very complicated system, that's why they calculate this phi parameter. So more complicated system, more consciousness. But in their view, we cannot copy the human, let's say, consciousness into the computer, because you need a human body and human brain to produce this type of consciousness. The interesting part is that up to now, everybody is just having their own theory and not talk to each other or not, let's say, confront each other. And a couple of years ago, those two groups, they established something called adversarial collaboration. So they are just trying to set up the same sets of experiments just to have an answer, which side got the stuff right. But of course, it started the war, like 2 years ago, 2023. There was a preprint of a letter signed by over 124 prominent scientists. They said that this integrated information theory is just a pseudoscience and we should just abolish it. And yeah, so things are happening all the time. So theory is not very conclusive. If we take a look at the, so let's take a look what we can measure. What? 10 minutes? Oh. So it will be a little bit, yeah, can I just? Yeah, okay. Thank you. No one is sleeping, so I assume it's not that bad. All right, so they're going to be shorter for sure. Okay, so even if we take a look at this, what we saw just using this neural correlates approach, and I think even if we are not being able to reach the ultimate answer, we can find out a lot of cool stuff on the way. For sure, we know that even in a regular life, this is this green part, we got different states of consciousness. Like right now, a majority of you are, focusing on the board, but some person can be just, zooming out, thinking about something else, or you can, fall asleep. Which Monday morning is really natural. But also you got also a lot of different troubles with consciousness. Like for example, something that is not used already, but still in some literature, like a vegetative state means we see living organisms, but we have no idea whether someone is still in the brain. Or, for example, minimally consciousness state or different states that our consciousness is not working on 100% efficiency. Of course, we can really distinguish between the brain death and the healthy brain. That's a healthy brain with normal, the PET is just measuring the amount of glucose consumed by the tissue, so that's a healthy brain, that's a very deep vegetative state. But what about something between? And yeah, Piotr is here, so I'm really nervous about explaining all this EEG stuff. But so one of the techniques that was developed during with these experiments was zipping techniques. So, because in case when we have no contact with a person, you cannot ask about this level of consciousness. It's like, for example, asking someone really drunk how he feels, okay, or she, okay. So, in this case, this technique is something that you can measure the capacity, let's say, ability of the brain to process the information in indirect way. So they use a external magnetic field just to, let's say, like stroke the brain, just evoke the activity in many cells under the skull, and then they record the EEG, means how the cells are talking to each other. And we have no idea what's exactly in all the signal of EEG, but they realized that if you just try to compress this signal, when there is something interesting inside, the file is just big. If there is nothing interesting in a signal, the file will be just small. It's like exactly with a picture. If you just take a picture of the white wall, the compressed picture will be very small. If you just take a picture of this hole, it will be much bigger. So that gives the index of, let's say, complexity. And probing people in a different state of consciousness was really correlating with this index. So here we have vegetative state, minimally conscious state, emerging from this state, and evoked patient. And you can see there is a correlation of this artificial coefficient. And also, if you use anesthetics, you can really see that here is the coefficient of people being awake, and here is under, for example, when sleeping, midazolam, xenol, propofol means anesthetic. So that's the way the anesthesiologist will know that you are awake, even if you are paralyzed. So that's give us the hope. And also that's spring the new theories for theories of consciousness. For example, let's use psychedelics and go to the other way. Like if we have this primary consciousness over here, if we go here with anesthesia, so we are turning into someone who is less conscious. So maybe we can go to the other side, and in some very narrow window, we will just have a better consciousness. All right, but sometimes you have to go deeper, and because EEG got the signal which is very filtered by the skull and skin and so on, but sometimes you have access to the patients that got electrodes inside the brain. And this is right now, in many countries, standard procedure before the surgery for epilepsy, because in epilepsy sometimes you cannot see the reason, like the tissue looks like it's healthy, but there is some group of cells that are causing trouble, and you have to just remove them. So if you cannot see them, maybe you can use some triangulation and other sensors. In this case, you put electrodes directly on the cortex, or sometimes you just put needles through the brain and record for a couple of weeks the activity of the brain. And that picture is not from the horror movie, it's from the research paper about the linguistics, because they got a patient and they were trying to find out when you have a feeling that you want to say something, how long it takes to say something, okay? And in this case, there is one observation. The patient got an electrode in a claustrum. This is a special very interesting place in the brain. And because you got this electrode, you can record the activity, but also you can use a current to disrupt activity of the place of the brain. And this is just observation. It's not regular research that, yeah, I just put a, star. But in this case, the research was able to switch off the consciousness of this lady.
Have no memory.
She didn't fall asleep. She was just, like hung in the middle of the movement and not able to record any memory.
When Mohammed sends a 14 milliamp electric pulse into the claustrum, the patient freezes mid-sentence. It's as if her conscious mind has been switched off.
Cognitively, she...
All right, so this is just observation. We shouldn't conclude it could just spring other research, but this claustrum is really interesting because in mice we find cells that got dendrites, means inputs from whole the brain. This is a very interconnected structure. So maybe it's, of course, not a place of consciousness. Maybe it's just something like coordinator. All right. So speeding up, interesting cases. If we just take a look at what people were doing and how it connects with consciousness. For example, we got a patient with split brain. This is surgery that was performed on patients with pharmacology-resistant epilepsy. The idea was that if you got a site of problems in one of the hemisphere, if you just cut the hemisphere apart, cutting the corpus callosum, the biggest fiber in our brain, then the epilepsy cannot travel from left to right, and you will stop it in one's hemisphere. And yeah, that works. But in many patients, they got like two personalities, like two consciousness for a while, because other connections are sparse. So the person was able to, let's say, figure out how to live with two consciousness in one single brain. Because, for example, those patients right after the surgery were just, one hand was, because one hand is controlled by the opposite hemisphere, one hand was just, buttoning up the clothes, and the second one was just opening it, okay? Or just putting, for example, something from the shelf in a shop, and the second hand was putting it back, right? Also, how much brain do we need? This is a picture of the brain of this girl. Like half of the brain is missing. The half of the brain was removed because of the Rasmussen syndrome. This is unspecific inflammation of the brain. And if you removed half of the brain, cortex and subcortical parts, in an early age, you will have a normally functioning person, almost normally functioning person. And the oldest girl that got this surgery was in Poland, I think at the age of 13 years. That was done 2017. Also, you can find out very different and strange cases, like this paper in Lancet that was called the white collar brain, and the person came to the physician because of the pain in legs, and they realized that the person is got no brain at all. That was because of the hydrocephalia. It's a overproducing of the cerebral fluid inside the brain, and because it was so mild and takes like tens of years, his tissue was able to adapt to, first, loss of the volume, and second, the whole brain tissue was squeezed out over here. Here is just the fluid. And this is a case which is really mind-boggling, because that's kids that are interconnected with the brain, Siamese twins, and in their case they can see through other eyes if they want to. So of course this is, let's say, natural experiment, but this is really something like we can see that someone got a half brain or brain split, but here is a 1.5 brain. So what about their consciousness? They also share some consciousness with between them. All right, animals. With natural point of view is over here. We are humans. We are conscious. Other humans are conscious as well. This is our natural assumption. But philosophers say, okay, how do you know that? Maybe all other people are just, robots or just virtual reality for you. We got no idea how we can know that. And also, maybe we are all just protein robots, and this consciousness, as Daniel said, is something completely virtual. On the other hand, we can apply maybe our cousins, like great apes, maybe they got also some kind of consciousness. Maybe complex animals, but what does it mean complex? Okay? Maybe all living systems, or maybe all world is conscious. So even on such a scale, we have no idea where to put, let's say, the cutting point. And we really got a problem because animals are different. Their, let's say, embodiment of their life is really connected to the environment. So for sure we have to use a different, let's say, tasks and tests for mollusks like other mammals. If we just take a look at the brain itself, we still have no idea why our brain is so special that we produce the culture and are the most prominent species over here. In case of brains, we can see that you got huge brains which are not that very intelligent, and let's say the complexity is also, for example, density of cells per cubic centimeter is also very different. For example, here is the African lion and chimpanzee. You got exactly the same size of the brain, but much bigger density of cells. If you take a look at just a cortex, it's really hard to judge. Okay, that's a human one. If you don't got a ruler that will tell you about the size of the cells, the structure is very similar. So what about the size? So one of the measurement of the uniqueness of human brain is that it's so big for an animal of our size. Like we would expect for our weight 4 times smaller brain. Taking a look at the other species, we got like a 4 times bigger encephalization quotient. But we still see the similarities in the build. For example, this is a mice brain. You see cerebellum, which is mainly for a movement. We see spinal cord with the same structures, hypothalamus over here, hippocampus over here. So we see that there are similarities on the cellular level and on the anatomical level. But what about the uniqueness? And that was a really cool and very surprising paper, because what they did, they just take astrocytes. This is a cell, glia cells, that are helping the neurons. So we think that astrocytes are not processing the information; they are just to make the neurons, better. And they really got the job right, because researchers were transferring humans' astrocytes into the mice, and the mice were much smarter. So the smartness was not in the neurons; it was in the astrocytes. So that's really spring the whole set of research, and it was, replicated many times, and it really works. All right, and what about, the mammals, okay, fine, especially in the same, let's say, environment, that's cool. But what about, for example, some animals that are on a completely different evolutionary branch, like our common ancestor was really, like a no eyes and quite kind of not very intelligent. Then we got a really problem, because first we need to develop the tools that will, for example, deal with the complex, how the animals is able to process the complex information, and also what we should look for, okay? Because we have different anatomy and probably a little bit different cells as well. And one of the tests, which is really crude and rather for fun than for scientific research, is a mirror self-recognition test. The idea is that the animal should be able to recognize itself in a mirror for the first time, like without learning, because you can teach, for example, pigeon and dogs, but species that are able to do this on the first time are like chimpanzees, bonobos, orangutans, dolphins, elephants, I think just one case, and for example, European magpie, sroka. And that's surprised because the crow is not able to do so. And the test is you put a special, like a marker on animal. The animal is not able to see it on itself. It can be seen only in a mirror. And after you present a mirror, the animal should start to, trying to remove it, like a parasite. And the most surprising discovery was that this creature, the blue streak cleaner race, very small fish, is able to do so. And exactly those fishes are, marked to know which one is which, with small amount of silicone put under the skin. And one of them, when presented with the mirror, was starting, wrap itself on a stone. And they repeat this experiment many times, and this species is able to do so all the time. And it's really, this species is really special because it's, that's why Janek said I'm starting research fish, because it's really very small brain, living in a, like a 20 degrees tropical reef, which is much lower, let's say, chemical power of processing signal, but it's the behavior is really complex. So we are starting the project. We got this project for like 2 years, and we're building equipment just to do cognitive tests on such a on this fish. All right. The end. We have no idea what the consciousness is. We have no idea where to look it in animals. So we came up with Cambridge Declaration of Consciousness, that even if we got no neocortex or something that looks like a complex brain, we have to treat all animals as potentially being able to be conscious. All right, and at the end, new biology, organoids, because biologists like to play with living tissues. Probably you know that you can just culture the tissue in a dish, okay? So you give the tissue something to eat, and you got cells. For example, you can produce a grafts for a skin, and it's already established techniques. What about if you just let the cells produce something which is like a spherical, and in this sphere you have a different types of cells, and you took those cells, for example, from the fibroblasts of the patient, then you end up with some blob of tissue which got, for example, neural tissue, exactly like a donor, okay? So if it's small, it's not a problem. But researchers were able to keep such a development up to the stage when you can see, for example, forming of eyes, like in a fetus, okay? So, and the gene expression was exactly like in so you're just producing really like a cloning of the person on the small scale. Of course, we are not able to go further, like, forming the whole, fetus and person, but still, researchers were able to form such organoids from the patient with Rett syndrome. This is a genetic syndrome which is characterized with the epilepsy attacks, and they saw in neurons in such organoids the improper pattern of electrical signals. Okay, so why they do so? Because this is a perfect way to personalize the medicine. Okay, you don't have to test the drugs on the patient. You can build organoids, many of them, and then test drugs on those organoids and set the treatment for a person. So that's the idea behind those research. But those research go further. For example, this is a paper where such organoids from humans were implanted into the rat cortex. And those organoids were able to interconnect with the nervous system of the rat. And they use special optogenetics techniques with lasers, so they were able to switch on and off this graft. And the graft was controlling the wanting to drink. Last year, there were also a paper that was trying to connect a human organoid, human neural cells, to the computer, just to force those cells to compute something, which is really cool. And that's lead us to the last part, artificial intelligence, because one of the biggest questions is, are we able to produce a system in silico that will be conscious, that will say, okay, I am I, okay? And the question whether we can do so is still, the answer is still we have no idea. So, of course, we can ask ChatGPT what's the artificial intelligence. It will say very gently that this is just a computer science, branch of computer science, and that should mimic our abilities. But I think we already reached the moment that was predicted by Arthur C. Clarke, that any sufficiently advanced technology is indistinguishable from magic. And I think that's exactly the case. And that will be a problem, because since we cannot recognize the consciousness or proto-consciousness in animals, we are not able to do so in a computers. But just to make like a bracket, we start with the Nobel Prize. We will end up with a second Nobel Prize from last year. It's for Baker, Hassabis, and Jumper. And Hassabis and Jumper, they developed a system called AlphaFold. And this is, of course, system based on the deep learning neural networks. And this system solved the problem that was one of the biggest in biology. Like, we know that proteins are comprised of amino acids. We know the sequence of amino acids, but the protein folds itself, makes 3D structure. And we got no idea how to predict this structure. Okay, so of course we can have games, but also we can have competitions, and scientists will try to compete with accurateness of predicting. And up to AlphaFold, the prediction rate was like a 40%. And right now, the AlphaFold 2, the system that was granted with the Nobel Prize, got it up to 90%. Right now it's AlphaFold 3 and it's got like 99% accuracy. So we still have no idea how it works, but it at least solved the, one of the biggest problems in biology. But then we got the problem, for example, like 3 years ago, one of the engineers from Google claimed publicly that the language model from Google, Lambda, is conscious, and he wants to be a sentinel of this model. Of course, that was probably some kind of like a hype, but there are already people who are really feeling that, for example, chatting with ChatGPT is much better than chatting with other humans. Okay, sorry for being a little bit over time, but if you are interested in the talks and these topics, I can really say that this series, Closer to Truth by Robert Lawrence Kahn, is really cool because he was exactly start with this question, like what is the consciousness? Can we got a research? What are the theories? And there is like a really huge series of interviews between 2000 and 2024, exactly about this question. Why consciousness is a mystery? So all the people that I mentioned in lecture, probably you can find them in this series. But the worst message is that this guy produced a paper last year and with no answer, just showing that in the meantime, he just calculates over 200 different theories of consciousness, and half of them are, let's say, neuroscience and science based. So we'll have a lot of stuff to do in the following years. Thank you very much.
Questions, please.
So if you try to compare the complexity of human brain with the best AI systems nowadays, so if you just look at the connections, I think it's a factor of 100. Yes, that you mentioned we have 100 trillion connections. In, I think, best AIs, it's now 1 trillion. So would you say that it's just a factor of 100 that we need to make like a structure that is equally complex, or you would think that it's more about all this physical background that it's inside that changes the behavior of the system? It's not only the number of connections that's relevant.
My feeling, and that's a feeling, is that not only about the connections and the power. And people in the field, they really speaks from rather the feelings than the experience. But I think the first aim is the question, are we able to produce a human kind or type consciousness in a machine? And my feeling after doing a lot of reading and research is like no, because to produce a human type of type consciousness, you need a human being that is like an entity, embodied, got a history, was born, got a lot of social interactions, okay? Because, for example, one of the biggest thing that our brain got is the will to survive, okay? Even if we are, lost in our life, we still have to, eat, protect ourselves, and so on. And this is something that is not dependent from amount of connections, okay? Because you can see such a pattern even in this fruit fly with just very small amount of connections. So I think it's rather something which will be qualitative than quantitative, okay? But I cannot prove that.
So I think that the main problem is what is conscious and which is not. And I think one of the theories I heard was the attention, that what we are performing with attention put to it, it's conscious. So it needs some top-down information coming. Like we can perform very complicated actions like walking. Okay, walking is quite complicated, but it's not, we need not consciousness to perform it. So maybe it's similar to artificial neural networks. It can perform quite complicated actions like, well, similar to walking. Now it's really a problem also with robots, if they can walk, but it will not be conscious all the same. So it's no matter of complication of the action, but the attention of the intent to be aware of performing given action.
I concur, because, but of course there is a problem that in case of even other humans, yeah, what we can see is just a behavior, or I don't know, report that okay, I'm feeling this, I want to do this and this. So there's always this barrier that we are just seeing action, and if we just probe, for example, animals, we can see, okay, I scared the rat is just running away. But I can build a robot from Lego bricks, so I make a huge sound and the robot will escape. So what is, I can put a fur on it. We did, because we were trying to scare the real rats with the robots. So you have a system that you can see behaves exactly as you are expecting, and maybe this robot also got something attention, like a special loop in a software that is just, you know, looking and scanning the sensory input where there is some kind of stimuli. And if I can name it like a consciousness, I don't know, that the robot is just, let's say, aware of the environment.
I don't mean attention, that like a perception, you know.
But attention like my will to do so.
Like intention to do so, and that's something which in brain is going top down. Okay, so maybe in the cortex, in the frontal cortex, there is maybe not a circuit, but probably a complicated system that somehow is steering this attentional behavior.
So exactly, I think that's the question behind these theories, that if there is a place, a small number of cells that are, you know, having this attention and just focusing, or it's a network, or it's just a collaboration. Yeah, for sure, but how big? Yeah. Yeah, that's the question. Yeah.
I wanted to ask about the statement you made at the very beginning of the presentation about the proto-morality that is in it. So I wanted to ask, what are the premises or proofs for such a hard statement that solves years, millennia of philosophical inquiries and questions, basically?
Experiments. Exactly, experiments. That's why I like this neural correlates approach, is that you stop philosophizing and thinking how it is, you just start to probing the reality. And exactly this morality comes from the research of Paul Bloom and Karen Frick. So they were trying to look for proto-morality in a very young kids, like couple months old. So kids that are not able to be influenced by the culture or be kind or be good, but you just present them with a stimuli, very simplified, like a puppet, and then you can ask question whether you like the puppet which is good, or you like the puppet which is bad, because the puppets are cooperating or, you know, fight each other. And it's really common that kids are choosing the puppet which is helpful, which share, and that is eating exactly the same food as me. Means is from the same tribe. If it's from the same tribe as me, then it could be nasty to other puppets. Exactly on the young kids. Yeah. And they like those experiments, yeah.
I would like to say that comparing the number of connections is very misleading, since a cell has also expression of genes, which is a very dynamical process in every cell, while comparing it to teaching the AI neuronal, let's say, net, is just changing the weight, which is orders of magnitude less complex than actual response of the cell for the changing environment. So expression of genes, which is not known up to now very well, while it happens at the given time in given environment, is making the problem extremely complex in comparison to any AI, even with the same number of connections. So it's not the whole story, it's the number of connections.
That's for sure. Yeah, that was my idea, to show that this is a dynamical system. And also we all got these chemical, receptors, because in case of artificial system, we just model, for example, the weight of connection. But in living system, we have these receptors which are different. For example, being more prone to the addiction. Okay, we know some people who can abuse the substances and be fine for the whole life. And others who just tries once and they hook up, okay? And in many cases, it's just the differences between the receptors, chemicals, which are based on genes, okay? So of course, we got much more bigger variability and sources of variability in living systems than just connections, that's for sure. And also, for example, we still have no idea how the really long-term memory works. And there are ideas that this is not rebuilding of the synaptic connection, but just rebuilding inside the cells, that the cells are somehow in a proteins keeping the memories. The memories are spread between many cells, but the changes are not on the synaptic endings, but just inside. So for sure we will find out the new phenomena that will lead us how the brain works. But for sure it's not just connections.
Last question, please.
I would like to ask about the organoids. What is currently stopping our development of these structures, and what are their complexities? I mean, what is the number of cells and what is the number of connections, and are there any ethical issues? Thank you.
Probably when I start preparing the lecture, those numbers will increase, because it's a very dynamic field. I think that the biggest achievement 2 years ago was keeping the mice organoids, which were really resembling the fetus, like up to, I think, 7 days, up from the 21-day pregnancy in mice. I might be wrong with the numbers, but it was like a huge advancement, and it was done by the Polish group in Cambridge. But if you are thinking about what is the constraints, it's changing, because the problem is that we have no idea how it will work, so researchers are really careful not keeping them too long in a dish, because they can start thinking and plotting against us. That's always the case.
Okay, let's thank the speaker again and see you in March.
Thank you very much.